Hi guys, this is chapter 12 and it's about the neural tissue. So this is the start of nervous system. There is 5 different chapters for the nervous system, and this is discussing histology of the nervous system, the organization in the nervous system, and there's some functional parts of the nervous system. So it's a good chapter to know before we go to specific parts of nervous system. Okay, so this slide is showing the general organization of the nervous system. So the nervous system is divided into 2 parts. The first part is the central nervous system, which is made of brain and spinal cord. And the second part is the peripheral nervous system, which is outside of the central nervous system. So the peripheral nervous system is made of the nerves. There are 2 types of nerves. One is the spinal nerves, which is connected to the spinal cord, and the second one is called cranial nerves, and that is the one that is connected to the brain. The second one is called ganglia, and ganglia is a collection of the neurons that are located outside of the nervous system. So neuron here, I'm talking about the cell body of the neuron, not just the axons, okay, or the branches of the nerves. The peripheral nervous system, based on its function, there's 2 different types. One is called the afferent system. Afferent system is going toward the central nervous system and is carrying sensory information. To get the sensory information, the nervous system requires receptors. Receptors are similar to receivers. So there is different type of receptors are there. Okay, so let's go over this one first. This is the somatic one. Soma means body, and this is the one that is mainly carrying conscious sensory activities. It is from the skeletal muscles, joints, and also the skin. So you may think, okay, how come a skeletal muscle, that should be motor, but the skeletal muscle also has sensory information, and when the time comes, I will talk about it. The other one is called visceral sensory receptors, and these are from the organs like heart, lungs, urinary system, gastrointestinal system, genital system. And then is the special senses. There are 5 special senses: sense of smell, taste, vision, balance, and hearing. So chapter 17 is discussing these special senses, so I will leave that for at that time. So right now I'm not discussing those things. So the information from these parts of the body, it goes toward the central nervous system. So that's sensory information. It goes to the spinal cord or to the brain through these nerves, and now the central nervous system is processing sensory information. It is important for higher brain function like memory, judgment, learning, personality, and also it processes motor information. So the motor is now leaving the central nervous system. This is why it's called efferent system. So the motor system is divided into the somatic and autonomic. Somatic, things that are conscious, and that is skeletal muscle. For example, if you want to walk, if you want to write, if you want to talk about something, that is the skeletal muscle. So you consciously decide and you do that. The second one is autonomic one, and the autonomic one is subconscious. So it is active even if you don't do any kind of activity. And that is divided into 2 parts. One is the parasympathetic, and the other one is sympathetic system, and they are supplying smooth muscle, cardiac muscle, glands, adipose tissue, and also lymphatic tissue. You can add lymphatic there, and the balance of the 2 is important for homeostasis. This is the histology of the nervous system. The nervous system is divided into neurons. So neurons are the ones that are carrying electrical activity, and there is another part that I will discuss it in a few minutes. Okay, so here is the neuron is divided into three different parts. The first part is these branches, and these are called the dendritic branches, or dendritic. These smaller ones that you see, these are called dendritic spines. So these are the branches and these are the spines. The function of the dendrites is to receive information. That receives information. And then is the cell body. Okay, so the cell body is called soma, and it contains a nucleus and nucleolus, very active. It also contains rough endoplasmic reticulum, and it has a lot of free ribosomes. So the combination of these two together under a microscope, it looks darker, and these are called nestle bodies. So these dark area that contains RER and free ribosomes. It also has Golgi apparatus, and there is also other filaments like neurofilaments and neurotubules. So the cell body is processing the information that is received from the dendrites, and then it sends it to the axon. By the way, the cell body has a lot of mitochondria because it's very, very active. Very active. The nervous system is processing, the central nervous system actually processing 20% of the body energy, so it's very active. So then it's connected to this long extension through this area that's called axon hillock. So this is the axon, and that is sending the information, okay? And that's connected through the axon to cell body. And the cytoplasm of the cell body, by the way, is called pericardium. So the axon is covered by a plasma membrane, and that's called axolemma. At the end, the axon branches, and when it branches, these are called collaterals. So these big ones are called collaterals, and then they branch again and become smaller, and these are called telodendria. Telodendrian is the singular form, telodendria is the plural form, and then they are connected with a postsynaptic cell. So synapse is the connection between the two cells. And this one can be a muscle or a gland, or it can be another nerve, so it can be anything. And there's also two different type of directions. So there is material that are made in the cell body, and it goes toward the telodendria, and that's called anterograde. And there is proteins that carrying that information, and that protein is called kinesin. So there's neurotransmitter that are chemicals that are made, and they are sent initially here. And there is another movement that are there that is going backward, that is called retrograde, from the axons, end of the axon goes to the cell body. So the retrograde one is carried by these proteins, and dynein is the name of the proteins. When a nerve is stimulated, so the direction is from cell body toward the end of the axon. So it receives information, it processes this information here, and then it sends all the way to the end of the axon. So it is a one-way direction. So what is a synapse? So this area that you see, this is the end of the telodendrian, and this area that is enlarged, this is called synaptic terminal. The synaptic terminal contains, endoplasmic reticulum, mitochondria, and also these vesicles. These vesicles are called synaptic vesicles and contains a chemical, and that chemical is called the neurotransmitter. So then this end is covered by plasma membrane. So this area is called presynaptic membrane, and then there is a space here That is called synaptic cleft. This is space. And then is another cell membrane, and that is called the postsynaptic membrane. The postsynaptic membrane here. So this postsynaptic membrane is part of another cell. So the synapse is the connection between the two cells. So it can be one neuron with another neuron. It can be one neuron with a gland, or one can be neuron with a muscle. So it depends on the location of the body. There is a different classification of the neurons, okay? So one is called anaxonic, so it does not have an axon. And you can see the dendrites and the cell body here, and that is found in the brain and special senses. The second one is called bipolar neurons, and it has one dendrite, okay? So it's small, that's connected to the cell body, and then is a short axon. So this is found in the special senses, and when we get to the special senses, I will remind you at that time. Then it's called the unipolar or pseudo-unipolar neurons that starts with the dendrites, and then this whole thing from here all the way to here is the axon, and axon is connected somewhere in the cell body. So this one is in the sensory neurons. So when we get to the sensory neurons, I will give you the examples at that time. This one is really long. For example, a neuron dendrites can be in your big toe, and the cell body can be near the spinal cord. So that can be that long. And then is the multipolar that have many dendrites, different dendrites are there. The cell body is processing it, and then has one big axon that branches into smaller teledendria, and you can see that in the central nervous system, and also a connection between a nerve and skeletal muscle. So now is the functional classification of the neuron, okay? So that was the slide before was how it looks under the microscope, and this is a functional classification. So there's three different types of neuron. One is sensory, the other one is motor, and the third one is interneuron. So the first one, the sensory, the function, it can be, depends on the location. It can be in the visceral organ, or it can be in the sensory organs that are consciously you feel it. For example, the somatic sensory will be in the skin, in the joints, and also in the muscle. How does it look like? They are unipolar, so it means that the cell body is located near the spinal cord, and dendrites are somewhere in the skin or in the muscle or in the joint, and then the information is sent to the spinal cord. There's also some of them are in the cranium, in the face area. Those are called the cranial nerves, but the majority of them are in the spinal cord. So there is three types of receptors in the sensory neurons, three types. The first one is called anti-receptors. So these are the ones that are receiving from the organs, for example, respiratory, cardiovascular, urinary, reproductive, and digestive. And there is also some of them are for special senses like taste and sense of smell, and the other ones are for the general somatic, which is deep pressure and pain. So they are located deeply in the body. Exteroceptors are located in the skin, for example, for light touch and pressure and temperature, or it can be for special senses, for example, sense of vision and sense of hearing. The deeper they are located in interoceptors. The shallower they are in exteroceptors. So touch and light pressure are lighter and more superficial, and the deep pressure and pain, they are located deeper in the body. And the third one is called proprioceptor, and these are the ones that are in the skeletal muscle and joints and the tendons, and they are important for the position of the body and also provide information about the movement of the joints. When the muscles contract, then the joints move, and that information is sent to the brain. Then is the motor neuron. The motor neuron again divides into two. One is the somatic one, and again, somatic is conscious, so that is the skeletal muscle. And the second one is the subconscious one, which is the autonomic or visceral system. So this is going to the smooth muscle, cardiac muscle, glands, adipose tissue, and lymphatics. They are divided into preganglionic and ganglionic fibers. So preganglionic means that the cell body is located in the central nervous system, and the postganglionic means the cell body is located outside of the central nervous system, and they are synapsed somewhere outside of central nervous system. So when we get to the autonomic nervous system, chapter 16, I will give you more information about that. Just for now, know it is two types of axon. One is preganglionic and the other one is postganglionic. The third type of the neuron is called interneurons. So it means that they are connecting the sensory and motor, and they are located mainly in the brain and the spinal cord, but some of them are also located in the autonomic ganglia, which is usually outside of the central nervous system. For example, they are near the heart, they are the lungs, near the gastrointestinal system. So those are the ones. Also, interneurons are located inside the walls of the gastrointestinal system, and that's a different nervous section that they are called enteric nervous system, enteric. So that is related to the function of the gastrointestinal system. So it's a little bit different. They are processing sensory information and sending motor information. So the motor response, for example, something is touching your skin and that's painful, and you are removing that part of your body, that is the motor response to sensory stimulus. Or you just want to walk or run or bike or something, so that's the motor activity that you want to do. So the interneurons are doing that, but they also are involved in other functions. They are called higher brain function, like memory, learning, planning, judgment, personality, emotions, reward system. All of those things are related to interneuron. So there is a lot of different functions about interneuron. Okay, so after that is you have other type of cells, and these are called neuroglia, and neuroglia are important for support of the nervous system, but the thing is it's not going to the next slide, so I don't know why it's not going to the next slide. Okay, good. So here you have the neuroglia, and there is two types. Okay, one is for the central nervous system, and the other one is for the peripheral nervous system. So you have four of them for the central, two of them for peripheral nervous system. The first one is called astrocytes. Okay, so it has a lot of branches. You see these astrocytes, there's so many branches, and they go and wrap around the blood vessels. Wrap around the blood vessels here. They are also connected to the neurons, so it has other functions as well. But the main function here is that is creating a wall or a barrier between the blood vessels and the interstitium. This is interstitium or extracellular fluid of the nervous system here, okay? So it is separating the content of the blood from the extracellular environment. So they are very different from each other. And that wall is called blood-brain barrier. Okay, so it's like a wall, it's not a real wall. It is also important for repairing damaged tissue. So if the nervous system is damaged for any reason, it repairs it. Unfortunately, the nervous system is not dividing. That's the problem with the nervous system. It does not have centriole. It doesn't divide. There's a couple of exceptions there. One is in the nose, and the other one is in the hippocampus, which is responsible for memory. But other than that, the nervous system or the neurons, they do not divide. So if they are damaged, then the astrocytes come and replace them, and that's why if somebody has a stroke or any other damage like a trauma or something, then their damage is permanent. If it is not for a long period of time, it's usually permanent. That's because astrocytes are preventing the axons to grow. So the axons cannot connect with each other, and the neurons will not divide. So that's why the symptoms and the signs are permanent if somebody has a stroke or a trauma or something. Sometimes if there's not total damage, the neurons, the axons actually grow, but they grow very slowly. But if there's severe damage, nothing can happen. Astrocytes are also important during the embryonic development of the central nervous system, and also for the 3D framework of the nervous system. So it has a lot of different functions. Oligodendrocytes, it means they have a few branches, okay, and they are wrapping around the axons, so look like hot dogs. And once they are wrapped around the axons, they create a myelin sheet. Okay, so this myelination is actually a good thing because that increases the speed of the movement of electrical activity. So the myelinated axons are transmitting information a lot faster. The third one is called microglial cells because they are so small, but they have a lot of branches here. They work like phagocytes, so if there is any kind of particles, any kind of tissue damage or any kind of infections, they go to the area and they clean up the area. So they look or function the same as phagocytes. And the fourth one is that epinemal cells, and these are some of them are flat, some of them are cuboidal shape, and they are located in the cavities inside the central nervous system. If the cavities are in the brain, they are called ventricles, and if the cavity is in the spinal cord, that's called central canal. So they cover those cavities, okay? And some of them also have the cilia, and they produce a fluid that's called CSF, cerebrospinal fluid. And I will talk about that fluid when we get to chapter 14. So for now, just understand that it produces some kind of fluid. So now is the neuroglia for the peripheral nervous system. There's two of them. One is called satellite cells. They are also called amphicytes, and these are the cells. Okay, so you have these neurons. The cell bodies of the neurons are here, and these are the axons, and they are covered by these satellite cells. So it's protecting the environment of the cell bodies. So the extracellular fluid around the neurons are different than the extracellular fluid that is outside of the satellite cells. So this gives it a different chemical components. Then is the Schwann cells. So Schwann cells are similar to oligodendrocytes. They are also called Neurolemma cells because they wrap around the axons and they create these, these cover around the axons. So they are creating myelin sheath. And the myelin sheath is also called neurolemma. So this is why it's called neurolemma cells. So once you have this, the speed of the conduction increase. If you look at it here in these two images, so this is an axon, and also these blue ones that you see, these are also axons. This is a thicker axon, and it is wrapped around by a Schwann cell. Okay, if it is wrapped by one Schwann cell in one specific area, that is called what? That's called myelinated neuron. But as you can see, one Schwann cell cannot cover the whole axon. So you see many Schwann cells are covering one axon. Okay. If you compare this one with this one, this is different. So these are all axons, but they are very small. So multiple axons are covered by one Schwann cell here. This is still covered by a Schwann cell, but the coverage is not that good. This is why it's called unmyelinated. So they give some protection, but is not affecting the speed of the electrical conduction that much. So this is why it is unmyelinated. Okay, so the speed is very slow in this type of neuron compared with this type of neurons. And also Schwann cells participate in the nerve damage. The nerve damage is called Wallerian degeneration. It's a separate discussion, but they can help the axons to regrow slowly. So it covers the area and let the axons regrow and connect back if the damage is not too severe. So now is a different discussion that's called membrane potential. All plasma membranes have electrical activities, the movement of ions, but the ones that are important are neurons primarily, but also the muscles. Muscles also have electrical activities. So they respond to the neurons. There is three different type of membrane potential. One is the resting membrane potential. It means that the neurons are not stimulated. So nothing is going on. There is no stimulus there. There is no electrical changes happening. The second one is called graded potential. And in graded potential, there is a stimulus arrives in the area, so the membrane potential changes. Okay, so the graded potential happens in the dendrites, in the cell body, in the axon hillock, and also here in the postsynaptic membrane happens here. The third one is called action potential, and action potential happens in the axon, so it transmits a long distance, and also it can happen in the plasma membrane of the another neuron, but especially in the muscles here, so in the postsynaptic membrane of a muscle. If it is a neuron here, it will not directly happen in the connection, in the synaptic connection, because the end of this axon will connecting with the dendrites here. So dendrites will have a graded potential, and then again cell body, and then it can have another action potential. So if it is just a neuron, it will have a graded potential here, and if it is a muscle here, then it can have an action potential. So it depends on the type of connection. It will have a different type of membrane changes. This is something that you may know from Bio 1, but it's good to review it. So this is a plasma membrane, and if you remember it, plasma membrane is a semi-permeable membrane. The outside of that is called extracellular fluid, and inside is the cytosol. So this is the fluid inside the cell. The plasma membrane may have different channels here, and these channels are made of proteins, and you also have some other proteins attached to the plasma membrane. So what happens here, that outside you have a lot of sodiums, a lot of sodium ions, so they are positively charged, and a lot of chloride, which is negatively charged. Inside the cell, you have a lot of potassium ions, which is positively charged, and a lot of proteins that are negatively charged. Okay, so you have these channels, these are called leak channels, that allow movement of these ions. So normally the potassium is leaving the cell, and here is the sodium channel, the leaky channel for sodium, that allowing sodium to enter the cell. Why? Because inside the cell there is not a lot of sodium, and outside of the cell there is not a lot of potassium. But you have something else here that's called sodium potassium pump, or ATPase, that hydrolyzing an ATP to ADP and phosphate. So it uses energy, and what will do is that it brings potassium back to the cell, so 2 potassium back to the cell, and kicks out 3 sodiums out of the cell. So because of this thing that happens, you have a membrane potential, which is normally at rest when there is no stimulation, is around -70. Remember, when there is no stimulation, and the ions, the charges inside the cell and outside of the cells are a little bit different. Inside the cell, you can see this is negative here inside the cell, because potassium can leave, but these proteins that are really big, they cannot leave. So the proteins are negatively charged and remain inside the cell, and that's why inside the cell is negatively charged. And outside of the cell, there is a lot of sodium there, and some of the sodium can enter, but they are kicked out again by sodium potassium pump, and this is why outside of the cell is positively charged. Okay. So if you look at it here, you see that the sodium channel, and then in the next slide you will look at the potassium channels here, okay, and the potassium and sodium ions. So here the sodium ion, there's two things to understand. One is the chemical gradient, and the other one is the electrical gradient. So for the sodium, let's talk about the chemical gradient. So there is a lot of sodium outside of the cell, not enough sodium inside the cell. So what does it mean? It means that a lot of sodium try to enter the cell. It's a chemical gradient. What about electrical gradient? Sodium is charged positively inside the cell because of the proteins, it's charged negatively. So it's a high electrical gradient for the sodium to enter the cell. So the net sodium electrochemical gradient is very, very high. So sodium really tries hard to enter the cell. There's something else is here, okay? That is, if these channels are not there, and you allow these ions to enter or leave the cell, okay? So a lot of sodium that were outside of the cell now try to enter the cell. They try to enter the cell, okay? And once they try to enter the cell, the inside the cell become more positive, until a time that there is no movement of the ions outside of the cell or entering the cell. So there is a balancing of the movement, so there is no net movement, and that is called equilibrium potential. And for sodium, it is positive 66. Remember, the resting membrane potential, when you have these channels and you have sodium potassium pump, is negative 70 millivolt. But the equilibrium potential, if you leave the ions to move freely and then it balances out between inside and outside of the cell, it becomes positive 66. Now let's look at what's happening with the potassium here. So for the chemical gradient of potassium, inside the cell, a lot of potassium. Outside of the cell, not enough potassium. So potassium tries to leave the cell. What about the electrical gradient? So the electrical gradient outside of the cell, it is positively charged, and the potassium is also positively charged. So there is not a lot of electrical gradient for the potassium. So this is why the net potassium electrochemical gradient is small. It's not that high. Although inside the cell is negatively charged, okay, so the potassium is positively charged, so the potassium enters the cell. If you're thinking only about the potassium, but there's also sodium normally there, so this is why the net potassium electrochemical gradient is not as much, okay, but the potassium has about 25% more possibility to leave the cell compared with sodium. Why? Because the potassium channel is leakier than the sodium channel. So it allows potassium 25 times more than the sodium to leave the cell. And if you are thinking of the equilibrium potential for the potassium now, if you let the potassium to leave or enter the cell until it balances out, it will be negative 90 millivolt. So a lot of potassium will leave, okay? So inside the cell will be more proteins, because proteins cannot leave, so the proteins will be there, and that will create more negativity. So it will be negative. And if you compare that with the resting membrane potential, which is negative 70, so the equilibrium potential of the potassium is much closer to resting membrane potential than the equilibrium potential of sodium, which is positive 66. So why the resting membrane potential is not negative 90? Because some sodium still enter the cell, and that will reduce the negativity. It makes it more positive, a little bit more positive. So this is why you have negative 70 because of the effect of sodium. Here we are talking about one ion at a time, but in real life this is not just one ion. You have sodium and potassium together, and you have sodium potassium pump, and that's why you have the negative 70 millivolt. So there is a change in the transmembrane potential can happen, and that change can be because of the opening and closing of those channels. And once those channels are changing, the opening and closing of those channels will change. That is the effect of the permeability of the membrane. The membrane, the plasma membrane, is made of the lipids. So, and this is why the ions cannot pass through that. So the ions need channels to pass through that. So once those channels open and close, then the permeability of the membrane changes, and the transmembrane potential also changes, becoming more positive, more negative. So for that, you need to think of the sodium and the potassium channels, because there's other ones also there, but these are the big ones. So these channels are divided into passive and active. The passive ones are also called leak channels. They're always open, and the potassium tries to leave the cell, and the sodium tries to enter the cell. And as I said before, potassium leak channels are 25 times more possibility to allow potassium to leave the cell compared with the sodium channels. And the active channels are normally closed. They are called gated channels. If there is a stimulus, they open, the resting membrane potential changes, and then they close again. So they need ATP. The passive channels do not need ATP. Okay, so there is three types of gated or active channels are there. One is closed, but they are able to open. So it's like this, okay? They are closed normally. If there is no stimulus, does not allow any ions to enter the cell. And once you stimulate it, then they can open up. Then there is an open one that is allowing the ions to enter the cell. And the third one is the closed, but cannot open. So here is this one. So once the ion enter, after a while it will close, and this is a different gate, and it cannot allow more ions to enter the cell. So there's three different states for gated channels. The classes of the gated channels, okay, one is called chemically gated channels. So this is the plasma membrane, and this is the gated channel, which is a transmembrane protein. It has a receptor, and that receptor is responsive to a neurotransmitter. So it's acetylcholine here, just an example, is response to that. So if the acetylcholine comes here, which is a neurotransmitter, the gate opens up and ions enter the cell. And once there is no stimulus there, then the gate closes back. So it prevents movement of ion to the cell. The second one is called voltage gated channel. Okay, so here is at rest, there is no stimulus there. You have two types of the gates. One is activation gate, which is normally closed at rest, and the other one is inactivation gate, which is normally open at rest. So when there is a stimulus comes there, okay, so see that there is from negative 70 now is becoming negative 60, so it will open up, then the ions enter the cell. Once the inside the cell become really positive, see this is positive 30 millivolt, then the inactivation gate will close. This is inactivation gate and prevents further movement of ion to the cell. The third one is called mechanically gated ion channel. So this is a protein, this is the plasma membrane. If there is no stimulus, nothing happens. If you press it, okay, physically press it, that will cause a distortion of the plasma membrane. That will open up the channel, allowing ions to enter the cell, and once the pressure is gone, they will close back. So as I said before, the membrane potential changes when the permeability of the ions, the permeability of the plasma membrane changes, and that will allow the ion to move across membrane. So once the ions move across the membrane, the concentration of the ion inside and outside of the cell will also change. And for that, you can get two types of signals. One is called the graded potential, and in the graded potential, when the electrical activity happens, it will be with a short distance. Okay, it will not go to the long distances. Compare that with an action potential that it can travel to long distances, and I will give you more detail about that. And based on this changing in the plasma membrane, the information can be sent or received or processed. So this is all the information, you know, sensory, motor, all the higher brain function is based on the transmission of the electrical activities. So here we're discussing about the depolarization and hyperpolarization. So what those two means, okay? So this is the plasma membrane, okay, semi-permeable. This is extracellular fluid. This is the cytosol inside the cell. So once a neurotransmitter comes and attaches to a channel here, this is like a gated channel, and that will open up the sodium. It can also be calcium, but sodium here, let's talk about it, can come here. So when they come here, inside the cell become what? More positive. So normally before the stimulus, it was negative 70. Once the ion enter, it become less negative, okay, or more positive. So that is called depolarization. Entry of the positive charges inside the cell become more positive. That's called depolarization. But if you have a different situation here, so this is the plasma membrane again, there's two different channels here. So a neurotransmitter comes, and now the potassium leaves. The potassium is positively charged. So if the potassium leaves, inside the cell become what? More negative. Or a different type of neurotransmitter can come, and more negative ions like chloride enters the cell. And this will creating inside the cell more negative. Okay, in that case, normally at rest it was negative 70, but now inside the cell is more negative because positive charge left or negative charge entered the cell, and now is negative 80, so farther away from 0. Okay, so it became more negative. So now that is called hyperpolarization. Hyperpolarization, becoming more negative. We are always talking about inside the cell, okay, not outside of the cell, inside the cell. So now what is graded potential? Okay, so this is a plasma membrane of a neuron here. So outside is positively charged, inside is negatively charged. So a stimulus comes here, and that stimulus changes this plasma membrane potential. Depends on the type of the stimulus, inside the cell may become more positive or it may become more negative. Also depends on the stimulus. The stimulus may be small, okay, so it may not travel that much, or it may be bigger, so it may travel further away. But there is a cytosol is here, okay, and that will prevent movement of the ions further. So this is why one of the characteristics of graded potential is that it will not travel long distances. It decays. It decays. So it means it will fade it. It will not go to the further distances. So that is happening. Graded potential happens in the dendrites in the cell body, and also it can happen in axon hillock and postsynaptic membrane. Compare that with action potential here. So action potential happens in the muscles and axons, okay? So here at rest you have negative 70. If there is a stimulus comes here, and if the stimulus is strong, that can cause depolarization. Remember, depolarization means the cell become less negative or more positive. So if it goes above a specific millivolt, that varies from one neuron or one tissue to another, that is called the threshold. If it goes above that, then it will cause an action potential. If it does not go above that, it still causes depolarization but not action potential. This is why action potential is called all or none phenomenon. All or none. Either it happens or does not happen. So if it happens, then it must go above the threshold. So from negative 70, it goes all the way to positive 30. So there is 100 millivolt changes. And it also goes to long distances. Okay, it does not decay, and it can go from one area to another area, and that will transmit information. So for the action potential, you have the sodium channels and you have the potassium channels. Sodium channels have two types of gates, okay? So these are voltage gated channels, okay? We are talking about the voltage gated channels. So respond to the changing in the plasma membrane potential. One is activation gate. So normally they are closed. They will prevent movement of ions inside the cell. Once the stimulus comes, they will open up and the ions enter the cell. Normally, they are closed. Inactivation gate is just the opposite. Normally, they are open. Okay, normally like this, they are like this, they are open. Once the stimulus comes, the ions enter the cell, and once you get to 30 millivolt, positive 30 millivolt, they will close. And it will prevent further movement of the ions inside the cell. So the activation gate normally is closed. Inactivation gate normally is open. So it's just the opposite of each other. And the sodium channel, so this was the sodium channel, okay? Now is the potassium channel. The potassium channel has only one voltage sensitive gate. And normally it is closed and it slowly opens up. So the gated channels, as I said before, they require ATP. They are closed normally at rest. The leaky channel, they do not require ATP and they are normally open. So what's happening during an action potential? Okay, so at rest, this is the at rest, so nothing is happening here. So you have the sodium channel and you have the potassium channel, and you can see that the membrane potential is negative 70 here. You have the two gates for the sodium channel. You have the activation gate and you have the inactivation gate. Activation gate is closed at rest, inactivation gate is open. And for the potassium, you have only one gate, and that is the activation gate, which is closed. So when these activation gates are closed, it prevents movement of ions. So sodium cannot enter the cell, potassium cannot leave the cell. Okay, so there's nothing is going on, negative 70. Now there is a stimulus comes. Once the stimulus comes, the activation gate, this was the activation gate, open up. Inactivation gate is already open, so nothing is happening. So look at it here. The graph goes up, so it's becoming more positive until it reaches here. So this is the positive 30. So once it reaches here, then something else will happen, okay? But let me show you in the next slide. So, but for now, I mean the next image, but for now, look at here. Potassium, the activation gate of potassium is still closed. So when the ions, sodium ions, enter the cell become more positive. That's called depolarization. Once it reaches positive 30 millivolt, inactivation gate now closes. Activation gate is still open. So this will prevent further movement of ion inside the cell. And so the cell it cannot become more positive because no more sodium enter the cell. But here is now the activation gate of the potassium opens. So once this opens up, potassium leaves the cell. Potassium is positively charged. Once potassium leaves the cell, what will happen? Inside the cell become more negative. See? Now the cell is losing the positive charges. It's becoming more negative. So this is called repolarization. So then what happens that the sodium channel goes back to its resting state. So activation gate is closed, inactivation gate is open. But the potassium channel is still open. So it means more potassium leave the cell. And now inside the cell become more negative. If inside the cell become more negative than negative 70, that is called hyperpolarization. So that is the changes that will happen during an action potential. So once this happens at the end, then you go back to the resting state. So here is two other things that will be discussed here about the refractory period. Okay, so absolute and relative refractory period. So let me start from here. This is at rest, no stimulus is there, and inside the cell, the membrane potential is negative 70 millivolt. Stimulus comes here, okay, and that is strong enough that goes above the threshold. So negative 60, negative 55, negative 40, depends on the location, okay? It goes above this, and that will cause a depolarization. This is depolarization. So here, the sodium activation gate is open. So more sodium enters the cell. From negative 70, it goes all the way to positive 30. Once it gets here, the inactivation gate will close. Activation gate is open, inactivation gate closed. Okay, and then the potassium gate will open up. So now the potassium is leaving. Potassium is positively charged. Once it leaves, the cell become more and more and more and more negative. Okay, until it gets closer to threshold. So from here to here, that is called absolute refractory period. It means that if you have another stimulus, nothing will happen. Why? Because in this part, the sodium channel is already open, the activation gate, and it will allow sodium to enter. So even if you do any stimulation, there's nothing happening. Sodium is already entering the cell. But once it reaches here, the inactivation gate will close the sodium channel. So even if you have a stimulus, the inactivation gate will not allow more sodium to enter the cell. So this is why it's called absolute refractory period. So once it reaches around here, now sodium goes back to the resting state. Okay, so activation gate is closed, inactivation gate is open. Potassium is still open, and this is why it's becoming more and more and more and more negative. So now it's below -70. So this is the hyperpolarization area. This area is called relative refractory period. Why? Because if you have a regular strength stimulus, nothing will happen because it is farther away from the threshold. The cell is more negative, nothing will happen. But if you have a strong stimulus, then it can overcome the threshold and you can have another action potential. So it can happen, but you need a strong stimulus. And if you don't have it, nothing will happen. That's why it's called relative. So once it, it goes further, the potassium channel will close, and then you will get back the -70, which is at rest. So resting membrane potential will go back to the same state. And then the cell will wait for another stimulus. You have two types of axons here. One is myelinated and the other one is unmyelinated. Okay, so if it is myelinated, this myelination will cause an insulation around the neuron. So it means that the ions cannot leave the cell in that area. So if you have a channel, a sodium channel here, and this is activated, it allows sodium to enter the cell, this area become positive, okay? So once it reaches in this area, this is called internode, okay? You have to have another channel here. So when it reaches in this area, so this is the voltage gated channel, and now that the inside the cell is positive, so it will activate this area. So this channel will now open up, and this area become more positive, but these ions cannot leave the cell. And then it jumps from this area all the way to this area. Now this area should have another channel. So then this will activate, it goes to another one. So this will happen throughout the axon. So because it is jumping from one to another like that, okay, and these ions cannot leave the axon. This is why the transmission is very fast here, and that's called saltatory conduction, jumping like a frog from one to another to another. That's why it's a lot faster. So myelinated axons are thicker and they are faster. Compared with the unmyelinated neurons here. So here you have a voltage gated channel. This is activated. This area become positive. You have to have another channel very close to that. If you do not have that, these positive charges will be kicked out by sodium potassium pump. So you have to have another channel very close to that, and then that voltage gated channel become active. Once that become active, that small area, the membrane potential will change, and you have to have another voltage gated channel very close to that, and then that will become active, and this should be another one here, another one here. So these channels should be very close to each other, and every time that they open up, that takes time. So even if it is a millisecond, but it still takes time. So this is why it is the conduction is a lot slower in unmyelinated axons. And because they don't jump from one area to another area, like the saltatory conduction, this is why these are called continuous conduction. So there is a classification of the nerves based on the thickness of the nerve fiber. The group A is the one that is they are thick and they are myelinated. So they are important for transmission of the information that require very fast processing, and they can be in the somatic sensory like a skin, or a somatic motor like a skeletal muscle, or in the joints. So things that are for survival that are important, or for consciousness that are important, they are group A. Group B, they are myelinated, but they are not as thick, so the transmission is a little bit slower. So you can see it in some of the fibers that are carrying pain, for example. And group C, they are smallest in diameter, they are unmyelinated, and you can see it in an autonomic nervous system, so at a subconscious level. The synapse is the connection of the one neuron with another cell, and you can have two types of synapses. One is electrical, the other one is the chemical one. In the electrical one, which is less common, one cell is connected with another cell through a gap junction, and that protein in gap junction is called connexon. So ion pass from one cell to another cell, and that a few cells are stimulated by a neuron, and then the other cells are stimulated through the gap junctions. And a good example is smooth muscle and cardiac muscle. So all muscles cells are not connected to a nerve fiber. Some of them are connected. The other ones get the stimulation through the gap junctions. And the other type is called the chemical synapses. And here you have to have a receptor or a protein in the plasma membrane, and that protein requires neurotransmitters. So here, that depends on the type of neurotransmitter. You may get a depolarization that may end up in action potential, or you may get an hyperpolarization that will not cause an action potential. So it depends on the type of the neurotransmitter. So here you have the two types of the neurotransmitter. One is called excitatory. So in excitatory, when a stimulus comes here, that will cause depolarization. So that will bring the cell membrane potential closer to the threshold. This is the threshold, closer to that. So that increases the chance of having an action potential. While an inhibitory one moves it farther away from the resting membrane potential. So you see it's -70, now it's close to -80. So farther away from the threshold. So this will cause hyperpolarization, which means less likely to have an action potential. So inhibiting transmission of information. So now the different types of neurotransmitters, so that they are named based on the type of the neurotransmitter that are released, okay? So the synapses are based on type of neurotransmitters. So this is called cholinergic synapses, and here is acetylcholine is released. And they are located in the central nervous system, in the connection between the two neurons in the peripheral nervous system, connection between a nerve and a skeletal muscle, and also in the parasympathetic section of the autonomic nervous system. There is also some part of the sympathetic nervous system that have cholinergic synapses, just a few, not a lot. And this is something that you have seen it before, a different image, but same idea here. So this is a nerve ending, and this is the muscle here. So you should have seen this in the chapter 10. So here you have a nerve ending, so an end of an axon, and this is the axon terminal here that contains mitochondria and also these vesicles. These vesicles contain neurotransmitter, and in this case, these neurotransmitters are acetylcholine. So once you get an action potential comes in this area, what will happen there? That will cause movement of calcium inside the cell. Once the calcium increase inside the axon terminal, that will cause fusion of these synaptic vesicles to the presynaptic membrane. These ones come and fuses to the plasma membrane here. And that will cause release of these neurotransmitters in the synaptic cleft. This is the synaptic cleft. And then you have to have these channels here. And these channels should have a receptor for that type of the neurotransmitter. So once they come there, they are connected to the these gated channels. This gated channel will open up and will cause what? Movement of sodium inside the cell. And when the sodium inside the cell, that becomes depolarization and action potential later on. So the rest of these, once you have these ones, you have also an enzyme here, and that enzyme will break down acetylcholine, and then acetylcholine is moved back to the synaptic terminal and then recycled. So they are made and then they are ready to be released again. So the second type of the neurotransmitter, these are called norepinephrine. They are mainly in the adrenergic synapses. Adrenergic synapses are part of the autonomic nervous system and the sympathetic system. They are important for attention and focus, consciousness, and waking up. They increase body temperature, and also they are important for regulation of the pituitary gland. So they are located in the sympathetic nervous system, but also in some other parts of the brain. Then is the dopamine. Dopamine is important for the subconscious regulation of the motor activity, and also is part of the reward system. So in Parkinson's disease, the level of dopamine is decreased, but in schizophrenia and cocaine use, dopamine level is increased. The fourth one is called glutamate, and that's important for memory and learning. And the other one is called aspartate, and that is important for movement of the skeletal muscle. So any voluntary movement that you do, conscious activity that you do, the neurotransmitter are released in the cortex of brain. That's aspartate. Serotonin is another neurotransmitter, and that's important for sleep-wake cycle. The normal level is important for emotions, and if the level of serotonin is decreased, the person cannot go to sleep properly and go under anxiety and become depressed. Also, it reduces the body temperature. If the level of serotonin is increased, the person may get hallucination, and some of the illicit drugs like LSD increase level of serotonin. Histamine, there's two types of histamine. One is the central one, and the other one is the peripheral one. So they have different functions, but in general, it will increase vasodilation. So that is important during sexual activity. Also, if there's any kind of inflammation or trauma, they can be released locally, and that will cause pain. They will cause release of the pituitary gland hormones, and also important for blood pressure, which will cause vasodilation, which will reduce blood pressure. And GABA has an inhibitory function. It's one of the main inhibitory neurotransmitters. Another one can be dopamine. Dopamine can be excitatory or inhibitory, but GABA is the only inhibitory one. And that can be used to reduce anxiety. So some medications contain GABA that will cause reduction of the stress and anxiety. There's also neuromodulators. So neuromodulator can be in a different neurons that will affect another neuron. So they are a little bit different than the neurotransmitters because they have to be made. So they are not readily available like neurotransmitters in the vesicles. So when they are made, it will take time. So the release of that is a little bit slow, but once they are released, the effect is a lot longer than the neurotransmitters. They can affect the presynaptic or the postsynaptic membrane or both. And also they can be released alone or they can be released together with the neurotransmitters. So the main reason for the neuromodulator is to increase the effect of the neurotransmitters, but the effect is a lot longer. So the main neuromodulators these are neuropeptides that are affecting pain. So opioids are reducing the pain. There are 4 different classes of opioids: endorphin, enkephalin, endomorphin, and dynorphin. So they reduce the pain. So they are actually produced inside the body to deal with pain. And if there is any kind of pleasure, also they are released. For example, laughter, that will cause release of these opioids, and then you feel less pain. And substance PCE is opposite of that. So they are released in the area that are damaged due to any kind of inflammation or trauma or infection, and that will stimulate the nerve ending, and that information is sent to the brain, and that's how you feel the pain. So it's just opposite of opioids. Information processing is a different topic. So far we talked about this area, okay, this neuron. So stimulus comes here, stimulating the dendrites. Information is processed in the cell body, is transmitted through the axon, and here is the neurotransmitters are released. And now we are talking about this one. So what's happening in the postsynaptic membrane? So this is what will happen in this area. So this is the thing that we will discuss in the next few slides. So the postsynaptic membrane, okay, so again we are going back to the type of neurotransmitters and the channels. So you have here a plasma membrane, and you have the different channels here. You have the receptor and neurotransmitters comes here. That will open up the channel, and that becomes more positive, okay. So it can, so this ion can be a sodium or it can be a calcium. So the inside the cell become more positive, which means what? Depolarization, okay? Increasing the cell membrane potential, getting it closer to the threshold, getting it closer to 0, or making it more positive. So these are called excitatory, okay? Excitatory postsynaptic potential. The other one is different, so that makes the cell inside the cell more negative. So here a neurotransmitter comes, potassium leave, or chloride enters the cell. Inside the cell become more negative. So this is hyperpolarization, and these are called inhibitory postsynaptic membrane potential. Inhibitory, further away from the threshold. So if you get a lot of inhibitory postsynaptic potential, what will happen? That will go further away from the threshold. That will inhibit action potential. And opposite of that is summation, that you will get a lot of positive a lot of positive graded potential, or a lot of positive depolarization, I should say, and the combination of all of them together will increase the membrane potential, and that will cause an action potential. Not always, but it increases the chance of having an action potential. So you may get, one neuron may get multiple stimuli. Some of them may be inhibitory, some of them may be excitatory. So if the number of excitatory are higher than inhibitory, they accumulate. That's why it's called summation, and that will increase the resting membrane potential, causing depolarization and increased chance of having an action potential. So based on how they will accumulate, you can have the temporal summation or a spatial summation, which I will explain it in this image. So in the temporal summation, so they are coming back to back. So that related to time. So one after another, they have to be very close to each other. When they are very close to each other, the potential will accumulate and increase, getting above the threshold. Once above the threshold, it will cause an action potential. So this usually happens in the body. You do not get one strong stimulus. You get many small stimuli. And an accumulation of all of them together, that will cause an action potential. So that over time, but they have to be very close to each other. The other one is that multiple stimuli from different neurons arrive at the same time. When they arrive at the same time in the same location, very close to each other, that stimulate the plasma membrane that goes above the threshold, and that will cause the action potential. So if they are small, then many of them should be there. Okay, if this was only one of them, it may cause a depolarization, but it will not cause an action potential because it stays below the threshold. So if many of them comes, they will go, they accumulate and they will go above the threshold, which cause action potential. And facilitation is just a part of the summation. So here you get one stimulus, it can cause a depolarization, another stimulus, so that accumulates, another stimulus. So this is part of the temporal summation. So once it gets to here, that means this is called facilitated. Facilitated. So it means what? It means that increased chance of having an action potential if you have another stimulus. If the threshold is here, and this is so close to the threshold, you get a small stimulus that will go above the threshold, it will cause an action potential. If you get a small threshold here, it will not cause an action potential. So this is not facilitated. It has to be very close to the action potential. I mean, very close to the threshold. And that is increasing chance of action potential. So here you have one axon and it's connected with another cell, and that cell can be, depends on the location. It can be a neuron, it can be a muscle or anything. But in this case, you have a third neuron, and that neuron will affecting this neuron. So it depends on what is released from the third neuron, then the final outcome is a little bit different. So in here you have one axon, and once the action potential comes, the calcium enters and the vesicles will release the neurotransmitter, and that will affect these channels in the postsynaptic membrane. Okay, here you have a different neuron that will release GABA. GABA is inhibitory. Once GABA is released, the amount of calcium that enters the cell will decrease. If the amount of calcium decreases, there's less exocytosis of the these neurotransmitter vesicles, and less neurotransmitters are released in the synaptic cleft, and less of them are attached to these channels. So this cell will not be stimulated as much. If you compare that with this one, you have a different type of neurotransmitter, which is serotonin, and that can be an excitatory neurotransmitter. And here more calcium enter. When more calcium enter, more vesicles attached to the presynaptic membrane, more neurotransmitters are released, and those neurotransmitters are attached to these channels and stimulate the postsynaptic membrane a lot more. So it depends on the type of the neurotransmitter that is released from a third neuron, then the final outcome will be very, very different. And that's it. If you have any question, please let me know. Thank you.